Children, welcome back to the class. In the previous class, we have studied about the reproductive system, how the organisms reproduce sexually and asexually, and also about the human reproductive system, etc. And now we are going to the next chapter, that is heredity. So, what is heredity? First of all, let us define the heredity. It means that each living organism has certain characters. They are called as traits. So, transmission of characters from parents to progeny is called heredity. So, the characters are transmitted from parents to the children or parents to the progeny, next generation. That is called as a heredity. And the process in which the characters are passed from one generation to another generation is called inheritance. So, heredity and the process is called as inheritance. So, before 18th century, that means when Mendel started his work, before his work, the scientists had a vague view how how the characters are transmitted from parents to the next generation, how it is possible, what are all the things that are responsible for the transmission of characters. So, these were the questions they were facing. So, at that time, in the year 1980, 1957, a Christian monk who is called as Gregor John Mendel, he started his work with the pea plants. Means he was also very much interested to know how the characters are transmitted from one generation to another generation, what are all responsible for the transmission of characters. So, by that time, 1857, nothing was known about cell division, mitosis, meiosis, genes, chromosomes, DNA. Nothing was known about all those things. But they had some doubt. So something is there, something is responsible for the transmission of characters. So that Mendel wanted to find out, and so he started his work in 1857. And he was a Christian monk. He started his work in the churchyard itself, and he worked with the pea plants. So, why did he select pea plants to study the transmission of characters? How he wanted to know how the characters are transmitted from one generation to another generation. So he started his work on pea plants. So pea plants, scientific name is, botanical name is Pisum sativum. And why did he select pea plants? Why did not he select other plants or other animals or other living organisms? So there are certain features which drew attention of Mendel. So what are they? So pea plants are annual plants. That means they complete their life cycle within 1 year. That is one thing. And the flowers in pea plant are bisexual. That means they contain both androecium and gynoecium. That is male and female reproductive organs. And the flower structure is like that. It is very easy to self-pollinate or cross-pollinate artificially. That is another advantage in pea plants. And pea plant exhibited certain distinct characters which are easy to study. And hybridization can easily observed in pea plants. Hybridization means when two different plants with two different characters are crossed with each other, and the resultant plant, next generation will show the characters. That was very easy to observe. So that is why he selected pea plants for his experiment. And he did completely 7 years, and he worked almost with 10,000 pea plants. And complete 7 years he worked, and he did all his experiments in the churchyard itself. And he selected 7 characters in the pea plant which are distinctly observed. So what were the 7 characters selected by Mendel? That means color of the flower. Some pea plants had purple flowers, and some pea plants had white flowers. And position of the flower. Some flowers are axillary in their position. Axillary means the flowers are produced in the axis of a leaf, and some flowers are terminal, tip of the stem. And color of the seed. Some plants produce yellow color seeds, and some plants produce green color seeds. And shape of the seed. Some plants produce round seeds and some plants produce wrinkled seeds. And in the same way, color of the pod, some plants produce green color pods and some plants produce yellow color pods. And shape of the pod shape was inflated in some plants and constricted in some other plants. And length of the stem, so some plants were tall and some plants were dwarf, means short. So in this way, he selected seven characters in the pea plants, and he worked almost with 10,000 pea plants continuously for 7 years. And he found out what are all, how the characters are transmitted. And for his work, his work was not recognized during his lifetime. So after his death only, the other scientists, they did experiments and they found whatever the discoveries that were made by Mendel were exactly correct. So his work was identified after his death only, and he was called as a father of genetics. So that was the introduction for this heredity. And now let us see what Mendel had done. So we came to know that Mendel has selected pea plants for his experiments. So why? So certain good characters. What are they? Pea plants are annual plants which complete their life cycle in 1 year. And next, the flowers are bisexual. That is, they contain both male and female reproductive organs. And it is easy in pea plants to self-pollinate or cross-pollinate artificially. That is another thing. And next thing, pea plants exhibited certain distinct characters which are easy to observe, and hybridization can be easily seen in pea plants. So these were all the characters which made Mendel to select the pea plant Pisum sativum for his experiments. And then his work started. And first thing we have to know that I have already told you before this, in 19th century, nothing was known about genes, chromosomes, mitosis, meiosis, all these things. So he has given certain names. Trait means character. Now we are calling as character. He called them as a trait. Factors, now we are calling as genes. And alleles means pairs. And F1 generation, first filial generation, F2 generation, second filial generation. So these are all the terms used by Mendel. Traits, factors, alleles. F1 generation means first filial generation, F2 generation, second filial generation. Now, so let us see what Mendel has done. So first he selected one character. He has taken one character into consideration. What is it? Color of the seed. Some plants produce yellow color seeds, some plants produce green color seeds. Okay, and he has repeatedly self-pollinated this, and finally he got the plant which produced only yellow color seeds. And he had selected another variety, the plant which produced only green color seeds. Continuously for several generations it produced only green color seeds. So he called them as pure varieties. Pure varieties, that means there is no mixing of character regarding the color of the seed. So he had taken a plant pure variety with yellow seeds, and he had taken another plant pure variety with green color seeds. And he crossed, means he pollinated between the two colors. So what happens in the F1 generation, first filial generation, he got all the yellow color seeds. He got all yellow color seeds. There were no green color seeds. The plant produced only yellow color seeds. Okay. And next, he has taken the plants which were produced in F1 generation as parents in the next generation. Okay. In the next generation, again, he observed that 3/4 of the seeds were yellow in color, and only 1/4 is green in color. So, but this green color did not appear in F1 generation. So that was the question. What happened to green color in the F1 generation? How was it possible to appear green color in F2 generation? That was the question. And he has taken different characters, a tall plant, dwarf plant, and he has taken the plant which produces violet flowers, and the plant which produces white flower. So single character he has taken into consideration. In F1 generation, he was seen only one character. And in F2 generation, the character which did not appear in F1 generation appeared in F2 generation. So how was it is possible? That was the question. He did with all these characters. That means the color of the seed, shape of the seed, color of the pod, shape of the pod, and the length of the stem, everything. So in first generation, F1 generation, only one character appeared, and in the F2 generation, the character which did not appear in the F1 generation was seen in the second generation, that is F2 generation. So after doing all the experiments with selecting single character into consideration, Mendel hypothesized. So he proposed three laws. He proposed three laws. What are the laws proposed by Mendel? Law of dominance, law of segregation, law of independent assortment. What is law of segregation? What is law of dominance? What is law of independent assortment? So that we will see. So here, law of dominance, law of segregation, and law of independence. So here, if we observe here, what we see now we say that it is in diploid condition, meiosis will take place and gametes are formed, haploid gametes are formed. The haploid gametes fuse together during the process of fertilization, and the zygote is formed and it develops into a plant. So that we'll say. So here we can say that here the gametes have formed. That means here, if we see here, capital Y, 2N, diploid condition. This is small y, that is green color seed. And during the gamete formation, so they get separate, capital Y, small y. And they fuse together and they form, all the things are capital Y, small y, capital Y, small y, capital Y, small y. All these things are capital Y, small y, capital Y, small y, and capital Y, small y. Okay, so these are the things. Now, so next thing, in second generation, what happened? You can observe capital Y, small y, capital Y, small y. And in here we can see F2 generation, capital Y, capital Y, small y, again capital Y, small y, and small y. That means you can see two different types of three different types of chromosome genes are observed. That means here capital Y, capital Y, small y, capital Y, small y, and small y. Okay, so now he proposed that here he said that each trait is under the control of two factors. Now we say that each character is under the control of two genes. Each trait is under the control of two factors. That means the yellow color is under the control of two factors. Green color is under the control of two factors. And he called the pair of the factors as alleles. Now we are calling pairs. He called them as alleles. So here two factors, and here also two factors, and during gamete formation they separate, and the resultant will be like this. So here you can say in the next generation, that means in F2 generation, what happened? 3/4 are in what color? Yellow, and 1/4 is green. 1/4 is green. How it is possible? He said that each trait is under the control of two factors, and when the two factors are same, then he called them as homozygous. So when the factors are under the control of, when the character is under the control of two different factors, it is called as heterozygous. Are you able to follow? So when one character is under the control of same factors, under the control of same factors, it is called homozygous. When the one trait is under the control of two different factors, it is called as a heterozygous. And in heterozygous condition, the factors are under the control of, the traits are under the control of two different factors. The traits are under the control of two different factors. Here we can say capital Y, small y, capital Y, small y. In such situations, one character will dominate over the other character. One character will dominate over the other factor. That condition is said to be heterozygous condition. And here we can see this is homozygous, capital Y, and here you can see capital Y, small y, and here capital Y and small y. This is heterozygous condition, and this is homozygous condition. Are you able to follow? Homozygous condition, these two, capital Y, small y. And when one trait is under the control of two different factors, it is called as a heterozygous condition. So heterozygous condition, we can see capital Y, small y, capital Y, small y. When the trait is in heterozygous condition, one factor will dominate over the other factor. In heterozygous condition, one factor will dominate over the other factor, and the factor which dominates is called as dominant. It is called as dominant. And one factor is suppressed. The suppressed factor is called as recessive. The suppressed factor is called as recessive. So here what had happened in heterozygous condition, capital Y, small y. That means we are representing the character which dominates with the capital letter, and the character which is suppressed, the factor which is suppressed is called recessive. So here in heterozygous condition, this dominant character will suppress this recessive character when it is in heterozygous condition. So look here, here it is small y. That means both the genes, that means both the factors are same. So that is why the green color has exhibited. Here in F1 generation, yellow color is dominating the green color. That is why capital Y, small y. All are in heterozygous condition. And here the yellow color is suppressing the green color of the scene. That is why green color was not seen. It was a recessive character. So that is why recessive character we are representing with the small y. Okay. And so if these hybrid plants are taken and they are crossed, then in the next generation we find that 3/4 are 3/4 produce yellow color seeds and 1/4 produce green color seeds. How it is possible? Out of this, only one is pure variety, capital Y. This is homozygous. Homozygous, we can say dominant. It is dominant. And this is small y. It is homozygous recessive. This yellow color is homozygous dominant, and this green color seed is homozygous recessive. And here we can see heterozygous condition. That means a hybrid. That means it is mixture of yellow color and green color, but this yellow color is dominating over the green color. So this is in heterozygous condition. So though here the green color factor is present, the factor which is responsible for green color is present, it is dominated by yellow color. That is why green color is suppressed and yellow color dominated. So that is by doing such type of experiments with different characters. So he selected one character at a time and he observed what is the ratio of the organisms that are produced in F1 generation and F2 generation. In F1 generation, all the dominant characters were seen. But in F2 generation, 3/4 of dominant character and 1/4 of recessive character was seen. So by studying all these things, so as he selected only one character into consideration, it is called as a monohybrid. So what is monohybrid ratio? So in monohybrid ratio, we can see 3/4 are the plant which contains yellow color. Only 1/4 is green color seed. So that is the ratio is 3:1, which is called as phenotype. Phenotype means physical appearance. For outer appearance, 3:1. That means 3 yellow color, the plant which produced yellow color seed, and only 1/4. That is only one part produced this green color seed. But if we take genes into consideration, what happens? We can see 1:2, 2:1. What is 1:2, 2:1? 1 is homozygous dominant, and that is capital Y, capital Y. So we are representing dominant character with capital letter. And here heterozygous condition, capital Y, small y, capital Y, small y. That is two parts. That is only yellow color seeds are seen, but there is a factor which is responsible for green color is present, but it is not able to express. Why? It is dominated by yellow color seed. That means the factor which is responsible for yellow color. And here you can see both the factors are small y, small y. That means both the factors are responsible for green color. So that is why this green color seeds can be seen. So that is why you can say this is homozygous dominant. One part is homozygous dominant and two parts are heterozygous, and one part is homozygous recessive. So this is called as a monohybrid ratio. Monohybrid ratio means only one character is taken into consideration and the cross is made, and the resultant resultant characters in the F2 generation. In F1 generation, all the dominant characters are seen, but in F2 generation, we can see 3:1 phenotype, physical appearance, and if genes are taken into consideration, the ratio will be 1:2, 2:1. 1 is homozygous dominant, 2 is heterozygous, and another one homozygous recessive. So these are all the results of Mendel's observations. So based on this, he proposed 3 laws. What are the 3 laws? Law of dominance, law of segregation, law of independent assortment. So what is law of dominance? Law of dominance means when one trait is under the control of two different factors, one factor will dominate over the other factor. So I repeat, when one trait is under the control of two different factors, so then one factor will dominate over the other factor. So here yellow is dominating the green factor. Okay, that is called law of dominance. And law of segregation. Law of segregation means during the formation of gametes, the factors they will separate. Now we say that diploid organisms, diploid cells will undergo meiosis and haploid gametes are formed. But by that time they did not know about mitosis, meiosis, etc. That is why he said that during the time of gametes, what happens? The factors will separate. That is called law of segregation. And the next one, law of independent assortment. That means when the factors segregate, they will retain their own characteristic features. That means they do not mix with one another. That means yellow color seed is not mixing with green color seed, and in between the medium color is not formed. Yellow color is retained as it is, and green color is retained by the plant as it is. So that is why they will segregate law of independent assort. They assort independently. They do not mix the factors do not mix with each other. They assort independently. So these are the three laws. He hypothesized. He just imagined and he hypothesized the three laws, and which were later proved to be correct, and that is why he is called as a father of genetics. So law of dominance again I repeat because it's very important. Law of dominance means when one character, one trait is under the control of two different factors, genes, one factor will dominate over the other factor. That is called law of dominance. Law of segregation. During the formation of gametes, the factors will segregate, separate. And law of independent assortment means the gametes they assort independently, they do not mix with one another. That is called law of independent assortment. So in this way Mendel he proved how the characters are transmitted from one generation to another generation, taking pea plants for his experiment. Hope you have understood, and we shall see dihybrid ratio and also determination of sex in the man, how it takes place, that we shall see in the class. Hope you have understood. Okay, thank you.